Okay, and everybody sees the slide, okay?
Yep, looks
That
great.
looks good. Excellent. Okay, yeah, so I'm gonna be breaking up the question and answer section into three parts because I don't like talking at people for 45 minutes in a row. I think we can be a little bit more engaging than that today. So I'm going to first talk about background information, and I'll have a question and answer session for that just to make sure everybody is up to speed and on the same page. I'm going to have two different sections on different parts of my dissertation research, and it'll become apparent what those are. And we'll have questions after each of those parts. But without further ado, I'd like to say good morning to everybody, except everyone on the East Coast. Good afternoon to you. I'm out here in the Pacific Northwest in beautiful Seattle, Washington. And I am at the University of Washington in Seattle. I work with two PhD advisors, Adam Leache and Alejo Rico Guevara. And my dissertation research, I'm titling it Towards a Phylogenetic Understanding of Avian Iridescence. Let's begin by saying that everyone here is a mammal. And if you didn't know that, I'd like to see you after class. So mammals only have one type of pigment available to them for coloring their fur, their hair, their skin. You can check yourself. It's all melanin, I promise. Birds, on the other hand, have a huge range of pigments and structural colors available to them to color their skin and their feathers. And in particular, my research is on structural color, so you'll see it up there in the upper left-hand corner with the peacock. And I think that it's a little bit misleading when I show you that the melanins and the structural color here are separate because a lot of structural color, including the structural color that I study, is based entirely on melanin. What I mean by that is there are these tiny melanin organelles embedded deep inside parts of the feather. So we'll orient ourselves to a feather right now. So everybody has seen a feather, right? I hope so. If you have one handy, you can take a look at the center part of the feather. That's called a rachis. Those things coming off of the central part, the rachis, are the barbs. And further dividing these parts of the feathers, we'll call these barbules coming off of the barbs. And the structures that I study are visible in cross-section. They're embedded in the barbules. And so I can look at the very top part of these barbules, the part that is exposed to sunlight, to see these nanostructural arrangements of the melanin organelles, the melanosomes. So, what does this have to do with structural coloration, iridescent coloration that I study? Well, I'll walk you through this. Don't be afraid of the math. I just wanted to give you an opportunity to have a better understanding of the physics involved if that was interesting to you. So we start with sunlight, as many things in biology often do. Sunlight is going to bounce off of the keratin of the feather, first of all. Some of that light is going to go through the keratin and hit the melanin, the melanosomes, and it's going to exit the feather through the keratin. What this does, though, is uh, it allows some of the light to travel further. It literally travels further because it is traveling through the keratin. And when it meets the light rays that have bounced off the keratin, it's now shifted a bit. So it's not in the same it's not in the same phase as the other light rays, and so you can have a potential positive interference effect, and I'll show you some of that on the next slide. But we know this phenomenon so well that we can predict what color is going to be reinforced by just looking at the thickness of the keratin layer that the light is traveling through. So to figure this out, we have to calculate the optical thickness, which is just the refractive index of the medium times the thickness of the medium. And that equals lambda over 2, where lambda is the wavelength that is being reinforced. So it is all about that keratin layer. So one of the most important features, arguably the most important feature for the conditions for iridescence, is melanosome arrangement. Because if these melanosomes are far from the barbule edge, if they have an irregular distance from the edge of the barbule, then we're going to get pigment-based color. This just means that light is hitting the keratin, it's hitting the melanin, and it's being absorbed. So when you see like black feathers, brown feathers, gray feathers, it just means that light is not exiting the feather, it's being absorbed by the feather. On the other hand, if we have melanosomes that are close to the barbule edge and they have a really regular distance from the edge of the barbule, we get iridescence. And just to illustrate this a little bit further, when these melanin layers are really well organized, if they have regular spacing, then we get positive interference and we see an increase in brightness and saturation in a particular wavelength. But if organization is poor, then we can get negative interference in which these light rays are actually destructively interacting with each other. So just keep in mind that when you're seeing these bright colors coming off of feathers, it's because these nanostructures are arranged so precisely within like nanometers of each other that they're able to interact with light itself. It's just absolutely incredible. Okay, some important features include melanosome shape. So there's quite a bit of variety in the shape of the melanosomes that are used to create iridescence. I showed you the simplest one, these large round melanosomes, but they can be smaller, they can be flat. And what we think this does is create more surface area and fewer gaps for light to pass through so that there's more consistent melanin surface for light to bounce off of. Oh, and that increases the brightness and the saturation of the color. Another important feature that also increases the brightness and saturation of this color is the presence of air in the melanosome. So a lot of birds have figured out this neat trick to degrade the central part of their melanosomes to introduce air, which will enhance the iridescent coloration even more. So you're starting to get a sense for all of these different morphological axes. The last one I'll talk about is the number of layers, the number of melanin layers that is in these feathered barbules. And the idea here is that if the light is passing through the first layer, the second layer, you can catch those stray beams with more and more layers. Obviously, there's like diminishing returns at a certain point, but typically more layers means you're capturing more of the incoming light and creating a stronger iridescent effect. Okay, so I showed you a lot of different morphological types. I'll just note that iridescence is pretty much everywhere in the avian family tree. So not every bird has iridescence, but it's pretty surprising how many do, considering we tend to think that these birds independently evolved iridescence. So take a look at some of the diversity that's in the bird family tree. A lot of these birds are not closely related to each other at all, but they're still producing this effect with their feathers. Um, and okay, yeah, so one might ask why so many birds have iridescence, considering it might not even be an ancestral character. We think that iridescence has some pretty important ecological functions, including in signaling and physiology. So, for example, most famously, iridescence is thought of as a sexual signal. So, in this particular example, paper out of 2018, these researchers timed this flash display on the gorget of a hummingbird, the throat feathers of the hummingbird, with a corresponding sound display. And they saw that those two signals were coinciding within like milliseconds of each other, which is pretty incredible. So that's pretty good evidence that iridescence is used as a signal. Another use for iridescence might be thermoregulation. So it was demonstrated in this paper from 2021 that iridescent feathers are able to introduce heat to bird skin a lot more efficiently than any other form of coloration, including melanin-based coloration that is not structural. So iridescence has a lot of functions. It makes sense that birds might want to use it. Another thing that I'll mention is that birds, even though they have, many of them have iridescence, a lot of them use very different morphologies to the same end. So there is a ton of diversity in the forms that birds use to create this same effect. So some of them have the large melanin-filled melanosomes, like in this brown-headed cowbird in A. Some of them have thinner melanosome form, like in this Nicobar pigeon. And then we see some flat melanosomes, some hollow melanosomes. Birds really just run the gamut here. So the takeaway from this introductory section is that there are a diversity of morphologies and a convergence of function when it comes to iridescent coloration in birds. My focal taxon are the Strysores, and that just means hummingbirds, swifts, and their nocturnal relatives. So I've put a star next to taxa where we know iridescence exists. So it might not be surprising to you that hummingbirds have iridescence. Their closest relatives, the swifts and the tree swifts, also have pretty incredible iridescence as well. And then confusingly, the closest relatives to that group, the Apodiformes, are completely nocturnal, and we tend to think of them as having melanin-based coloration, not structural coloration, with the exception of the nightjars all the way at the base of this tree. So it's not in the literature that nightjars have structural coloration, but part of my work includes descriptions for the first time of iridescence in this group, the nightjars. And I'll show you some images that I took demonstrating this. Okay, so I'll just go through some of the main theoretical things that I think about, some of the main questions driving my research. First and foremost, is iridescence homologous in birds? And that's just to say, do birds have the same ancestral characteristics when it comes to iridescent coloration. So I just showed you that birds evolved these traits independently, which means they probably don't have the same common ancestor with iridescence. And that is further supported by the fact that there are so many different morphologies of iridescence present in the tree of birds. What I'm more interested in than morphological homology is molecular homology. So I want to know if iridescence can be considered homologous from a molecular standpoint, meaning birds, since they have common ancestors and they use the same genes to create melanin and keratin. I want to know if some of the same loci, some of the same genes might be involved in creating these different morphologies. And I can't do that without first figuring out what genes actually regulate these nanostructural arrangements. So that is another key question that I'll ask and try to answer during this presentation. And lastly, from the morphological standpoint, I'd like to see how the nanostructural arrangements of melanosomes evolve over time using this group, the Strysores. And one of my hypotheses is that there is an intermediate state called glossiness that might serve as a stepping stone from matte black coloration to the simplest form of iridescence. So glossiness is a structural color and it should be pretty familiar to most of us who have seen corvids, like this crow down here on the bottom right. So it's that sheen, that white sheen that you see on black feathers that doesn't actually have a strong color associated with it. So we are getting some of the light rays that are being reinforced, but maybe not a very consistent color. Uh, yeah, so one other aspect of the morphological data that I'm looking at are, sorry, is whether different forms of melanosomes evolve from one another, and if they do, what traits evolve from which other traits, and whether or not those state transitions are reversible. Okay, that was a lot of talking. It's at this point I'd like to have the first question and answer session, and then I will continue the talk by talking about the morphological aspect of my research. So go ahead and raise your hand or drop something in the chat if you wanted to ask a question.
Yeah, I wanted to ask, is there a record species with the most layers ever known?
I think we're going to have an answer for that someday. Right now, the nanostructural sampling hasn't gone through like the complete list of birds with iridescence. Um, the most that I've seen personally is like 6 or 7 layers,
Okay,
which is quite a few. I don't know how useful 7 layers is, frankly, for capturing.
the seventh one
Yeah, like
adds.
like I said earlier, it's like a bit of a diminishing returns thing.
Mhm.
Okay, Beth Rinke says, bit of a silly question. I disagree. I have an iridescent nail polish on right now. How does manufactured iridescence differ from natural iridescence? Yeah, thanks for that question, Beth. I am under the impression that iridescence is iridescence wherever it is. So you get the same exact optical effect from iridescence wherever it's happening in the natural world and even in the manufactured world. So, for example, in a soap bubble, you'll see that there are like different shimmering colors, and that's the exact same effect as well. Something called, oh, what is it called? Gosh. Oh dear, I'm blanking on the exact term, but that's okay. It's exactly the same effect where there are two layers of different optical densities that are creating this sort of like staggered reflective effect that is interfering positively in certain wavelengths. So, Beth, I wonder if the nail polish doesn't have a reflective layer that might be bouncing off the keratin in your nails. I agree, that is so cool. Nature is crazy. Are there any further questions before we continue the talk? I really want to make sure everybody's on the same page before we move forward.
I have one, but I want to make sure we also stay on time for the hours. Cool. And I was just so you're gonna show some new discoveries of iridescence in night jars. And I was wondering how common do you think that is across the bird phylogeny where there's undiscovered iridescence in species?
Mm. I want to say it's not very common, but that would be kind of ironic, right? Because if it's not well known, it means I
Yeah, I
probably
know.
won't know about it. That being said, iridescence tends to be pretty like eye-catching to humans.
Hmm.
Luckily, we have some of the same visual channels that birds do, and we can see some of their signals. So a lot of the birds that we know to be iridescent, it's because it's just so gosh darn obvious. In the case of the nightjars, they were hiding it pretty well. I'll show you the images, but it's very subtle. It's only visible under certain light
Hmm.
conditions, and it's only like a few feathers on the crown of the
Oh
bird.
wow.
Yeah, and because these birds are nocturnal, humans
Mhm.
aren't really interacting with them in the wild very frequently. It makes sense that it hasn't really been spotted until this point. And frankly, I've never actually seen these birds in the wild. The reason that I was able to pick this out is because I observed a museum specimen. Okay, is it okay if I answer one last
Oh,
question?
absolutely.
Okay. I've been curious from Alexis Powell. I have been curious about dull green colors as seen in some sparrows and whether they are accomplished by simple structural mechanisms like iridescence. Even though sparrows don't have obvious examples of blue, super whites are other examples of structural color. Anything known about this? Sparrows. Sparrows are like passerines, right? Are they perching birds? I haven't looked at the sparrows in particular, but if you're seeing like a green coloration that has that nice sheen in it, and the color and the intensity changes depending on the viewing angle, that's pretty good evidence that that's structural coloration. Um, and green tends to be iridescence, although not it might not necessarily be iridescence. Yeah, so it doesn't have to be blue to be iridescent. Iridescence can be any color at all. Hopefully that answers your question, Alexis. Okay, who's ready for the next part of the talk? I am. Okay, so I showed you some background on structural color, iridescence, melanosomes, and some of the questions that I'm asking in my research. I'm taking two approaches here to answer these questions. First one is a morphological approach, and the second one is a molecular approach. So for the morphological side of things, I sample feathers from museums. These are mature feathers, so they are completely done growing and they're dead tissue. It's just keratin. And to look at the nanostructures, I do TEM imaging, which is transmission electron microscopy imaging. I'll explain more about that in a minute. On the molecular side of things, I actually went into the field to collect developing feathers, so living tissue that still had RNA in it, so that I could do some RNA-seq, which is just sequencing of RNA molecules to see what molecules are potentially regulating these nanostructures. Okay, so to image an iridescent nanostructure, I first have to take the feather and embed it in some epoxy resin. So that's just plastic that gets very hard and keeps the feather nice and tight while I do some very thin sectioning of the feather. So, like the feather sections, to be able to see them under the transmission electron microscope, have to be less than 120 nanometers thick total, 120 nanometers thick. Okay, so these are pieces of plastic that are several times thinner than your hair, and I have to be very delicate with them to be able to put them under this machine that is larger than my entire body to be able to look at these structures that are nanometers thick. So here's some gratuitous images of me going through the motions of embedding feathers in resin. You can see I'm very excited about being in a lab coat. I don't get to do that very often. I look very scientific in that photo. So I took pieces of feathers that I wanted to look at, and I embedded them in these tiny pieces of plastic, like I mentioned. So this is under a light microscope here, and to get them to that thickness that I was talking about, less than 120 nanometers, I had to use a very fancy machine called an ultramicrotome, which is just essentially a glorified deli slicer. It is a very powerful microscope with this attachment for a glass knife. The first step involves whittling down the tips of these plastic blocks into these trapezoidal shapes. And to be completely honest, it was this step that was the most frustrating and difficult to master because it is completely by hand. I had to work with, and you can sort of see it in this image here. There's a loose razor there that I had to take by hand under a microscope and then create this trapezoid out of this plastic so that I could take thin sections from it. Um, it was difficult to master, and for a long while I was still learning how to do this, I was seeing trapezoids everywhere. But finally, I did manage to get the blocks into the right shape, and you can see all the way on the bottom right what the ideal conditions are here. So you have like this piece of the feather barbule or feather barb with the barbules coming off, and it's this trapezoid that I take the thin sections of using the ultramicrotome. Okay, so when everything goes well, I get to do the fun part, which is TEM imaging. So you can see I'm a lot happier about being in the basement with this beautiful machine, the JEOL 1400. Oh, she's a beauty. So, a transmission electron microscope is literally an electron cannon. It's the most sci-fi thing I've ever done in my life. It literally just shoots beams of electrons through these thin sections, these nanometer thin thin sections, and then what we see, the image that we see on the other end, are the shadows of whatever was embedded in this plastic. So on the image on the right, this is an image collected from the TEM. So what we see in the center here is the feather barb, and then on the sides are the feather barbules coming off of it. So the darker sections are areas where electrons were absorbed, and the lighter sections are areas where electrons went through. And on the lower left here, this is just a visual aid when I'm using the machine. I think it's cool. I wanted to show it to you because it's a phosphor screen that glows green when electrons hit it. So once again, the most sci-fi thing I've ever done in my life. That's what I'm working with here. And when I'm finally done at the end of the day, I get to see these incredible images of these nanostructures. So starting from the very beginning, here's the common nighthawk that I was talking about. You can see that it has some of these shiny feathers on its head, and you can tell how if this is a nocturnal animal, you would totally miss this. So here is a light microscopy image of one of the feathers. You can see it's kind of shiny green. And then when I look at it under the microscope, it's super clear that there are a ton of melanosomes. There's a ton of melanin layers here that serve to reinforce those light waves and create that thin film effect. That was the term I was looking for, thin film. And just to be super clear, light is coming in from the top here. So these nanostructures are interacting with light coming from the top. Here's another image, some more images of a different taxon. This is the crowned tree swift. Is that right? Yeah, I think so. The crowned tree swift. Um, here's a beautiful image of some of its really intense iridescence in its feathers. And you'll notice that not only are the barbules shaped differently in this bird, but the melanosomes have a different shape as well from Cordelis minor. So these are some of the flattened melanosomes. And this bird is a relative of the crowned tree swift. Is it the crowned tree swift? Crested tree swift. Thanks. This is the mustached tree swift, and you can see why. Uh, it is a very close relative of the other bird, and it also has these flattened melanosomes, although the melanosomes are a little bit less elongated than the other one. So I showed you these images because they're cool, but also because I wanted you to get a sense for some of the diversity of shapes and forms that I was looking at here. Okay, so one of my hypotheses was that glossiness is a putative intermediate state transition between matte black coloration and a simplest form of iridescence. I did this TEM imaging for dozens of species in the Strysaurus, spanning the whole group. I think in total I did about like 49 species. Um, And what I saw after plotting the traits on the Yet's phylogeny for all birds, and after doing some ancestral state estimation, was that my data don't actually support this hypothesis that gloss is an intermediate step between matte black and iridescent coloration. So looking at the tree here, the blue is glossiness, and it looks like in the taxa that I study, gloss is actually embedded in taxa that already have iridescence or already have this majority matte black coloration. So no dice there. Nonetheless, it was pretty interesting to see the morphological patterns in the group that I'm studying here. Uh, The implications being that either birds can evolve iridescence extremely quickly, straight from matte black coloration, or any transitional forms that might have existed in the past are now lost to us, and we might never know. Okay. One of the other questions I was trying to ask with the morphological data set was how these melanosome shapes evolved from each other. And I guess like the basic null hypothesis is that all transitions from state A to B to C to D are possible. And when I tested all of these different combinations of state transitions, what I got was a model that favors transitions to and from these large melanosomes and small melanosomes, but irreversible transitions from these small round melanosomes to either the filled, so non-air filled flat melanosomes, or the air filled flat melanosomes. Looking at the tree, it makes sense why this model might be supported, especially when we look at the hummingbirds here in pink, which all have this very derived flat filled or flat hollow form, and the tree swifts here in blue, which are the only group within the Strysaures to have these flat filled melanosomes. So, students of evolution might notice that there is exactly one transition on this branch from this green type, or the small round type that I marked in green, to the hollow flat type in pink, and another single transition with no reversals from the filled small type to the flat filled type here. So that means I don't actually have a lot of evidence on the phylogeny to support this model. Um, and it is based entirely on a single transition, essentially. Nonetheless, that is the pattern that I'm seeing in the Strysaures. And it's interesting to see in this particular group, although I don't think that studying structural coloration in the Strysaures has been super informative for the rest of the bird family tree. Still, nifty stuff in my unbiased opinion. Okay, so the next part of the talk is all molecular, and I have more fleshed out results there. We'll maybe take like 2 or 3 minutes for questions at this point because I only have like 15 minutes left. Uh, Regardless, I'd love to hear your questions about the morphological part of the talk.
Excuse me,
Yeah,
can
go ahead, no, no.
can I ask you a question about the phylogenetic tree and the morphological difference between birds, among
Totally.
birds? Yeah.
Yeah.
And so why do you think that different birds have different morphological melanosomes? Is there any kind of environment which forced the birds to have a specific morphological uh, morphological characteristics on their feathers?
Yeah. So, just to reiterate the question so I understand, you're wondering if there is like an ecological reason that birds have different melanosome shapes?
Yes, I wonder because, yeah, uh, looking at the phylogenetic tree you mentioned earlier,
Mhm.
I felt that the specific uh, bird groups had specific morphological characteristics. Yeah.
Yeah. So, personally, I don't think that the different melanosome morphologies are related to ecological factors, although I don't think that's been tested, so who's to say? I think that the different morphological characteristics are due entirely to historical contingency. That is to say, because these birds independently evolved iridescence, it makes sense that they would do it in their own unique ways, right, due to chance. Whether or not ecology or natural history affects whether birds have iridescence or not, I think there's more support for that. I think there's some recent work out of the Field Museum demonstrating that birds tend to have iridescence more often if they live in the tropics, for example. Thanks for that question.
Thank you.
Okay, I think I am gonna continue with the talk. Oh, I'm still screen sharing. That's good. Great. Okay. Yeah, so one of the questions, the main guiding questions for my dissertation is trying to figure out what genes regulate these nanostructural changes. And to do that, I sampled developing feathers from two species of hummingbirds that are present in the Pacific Northwest. So I didn't have to travel very far to do my research. Calyptiana and Selasphurus rufus. So the rufus hummingbird here and Anna's hummingbird here. So I sampled iridescent green feathers from both species and pigmented gray feathers from Calyptiana and pigmented red feathers from Selasphurus rufus. To get at the molecular data, I collected their RNA from these feathers and sent them to a sequencing facility where I got the raw reads and then did some bioinformatics magic to turn it into information that is digestible by people. So I've been wrangling data in R for 2 years now, and I'm still finding some pretty interesting patterns in the transcriptomic data. I also did nanostructural imaging for these two species. So in this particular case, I wanted a good morphological reference for what was going on in the molecular data. And for these, it's the same exact process as before. I just embedded a finished feather from museum samples and looked at it under a transmission electron microscope. So I have some fun photos of my field work here. So on the left, we have a hummingbird trap. So anybody who's familiar with catching birds in the field might be used to using a mist net, which is just basically just a long piece of fabric that birds can't see under certain light conditions that we use to just catch bycatch essentially. Hummingbirds don't fall for that, unfortunately, so we have to use more specialized techniques. In particular, we use this drop net trap that we bait using sugar water, which we know hummingbirds can't get enough of. And then there's this string that we pull that's not visible here to lower and lift the trap so that when hummingbirds are feeling secure and happy gulping up their sugar water, we drop the net on them and then they start freaking out and we have to pick them up and put them in a little sock to calm them down. Gosh, I should have taken a picture of the little sock. It's just so cute. Well, regardless, once we have the hummingbird in hand, I sampled the transcript or the developing feather and put them in individual little vials to make sure that there was no cross-contamination between feathers. I have some people from my lab to thank, including one of my advisors, Alejo Rico Guevara, and a postdoctoral scholar in the lab at the time, Jay Falk. So I didn't have to leave Washington State for any part of my research. And I got to enjoy some of the beautiful sights on the western coast of Washington, including on Vashon Island, where I camped for a day trying to catch hummingbirds by myself. So I think field work can be pretty exciting when you get to go to like fun new locations you've never been before. But in my case, I got to appreciate the natural beauty of the place where I was local to. Here's another gratuitous photo of myself doing lab work extracting RNA for the RNA-seq analysis. And now I'll show you some of the morphological results that I'm going to use to ground truth the findings from the transcriptome dataset. So for the rufous hummingbird, we see the iridescence that is typical of hummingbirds in the green feathers. This isn't new, this isn't novel. I'm not the first person to look at iridescent hummingbird feathers under a transmission electron microscope. We've actually had that information since the late 70s, believe it or not. However, I am the first person to look at non-iridescent feathers from hummingbirds under a transmission electron microscope, and some of the stuff I found was pretty surprising. So in the rufous hummingbird, I found that the melanosomes are made of a type of melanin that is totally different from the type of melanin that creates the structural colors. So structural colors are produced using eumelanin, and in the case of the rufous hummingbird, they use pheomelanin completely. They use all pheomelanin to create their red feathers. So the melanosomes produced by pheomelanin are irregular in their shape and size. I like to call them lumpy, morphologically speaking. And so it is pretty much impossible to create structural color from a pheomelanosome layer. The Anna's hummingbird, on the other hand, had a totally different pattern where we once again see the typical iridescent feather melanosomes in the green feather. And in the gray feather, I saw a never before observed melanosome type, which I thought could be reduced forms of the iridescent melanosomes. So in the gray feathers of the Anna's hummingbird, you see that the melanosomes are irregularly shaped. They have an irregular size, but some of them have these air vesicles still in them, and they are flattened as in the iridescent melanosome type. So this was pretty exciting to me because it told me that Anna's hummingbird, at least, might be turning their iridescent melanosomes into non-iridescent melanosomes or the other way around using a straightforward mechanism. So when I did the transcriptomic analyses to compare gene expression in the two types of feathers for each of the hummingbird species, what I got were a list of genes that were very highly expressed in one feather versus the other. So for Anna's hummingbird, comparing the green and the gray feathers, I saw that 1,224 genes were differentially expressed in those two types, and that is a lot. In the rufous hummingbird, however, I saw a tenfold decrease in the number of expressed genes, so only 126. And that was pretty confusing to me. Initially, I was worried that the data that I had collected for one species were contaminated or inadequate or degraded somehow. But the more I talked to experts and the more I looked at my own data, the more I started to consider that this was the actual pattern that I was looking at, which was pretty surprising to me considering that I thought the Anna's hummingbird feather types might be more similar to each other morphologically. Well, some of the genes that are most important to look at in the differential expression analysis are genes that differ in their expression by a twofold change or more. And in the Anna's hummingbird, I saw 65 of these genes, and in the Rufous hummingbird, I saw 10. So from this point forward, I'm not going to talk about the results of the Rufous hummingbird transcriptome analysis because most of what I saw didn't really have a relationship to color production or feather development. So I'm going to just talk about the results that I got from the Anna's hummingbird. And I think the most exciting one and most obvious pattern that I saw was that this gene, agouti signaling protein, ACIP, was had a sevenfold increase in expression in the gray feathers relative to the green ones. So once again, we get really excited about like a twofold increase in gene expression, and I'm seeing like a sevenfold increase of this one gene. To understand what agouti signaling protein is doing, we have to look at the melanin synthesis pathway. So here's like a super reduced form of that pathway. where we have a melanocyte on the outside here. So this is a cell type that produces melanin. And on the inside, we have the melanosome, which are the organelles that I was showing you earlier. So this is where the melanin is stored after it's produced. So MC1R is this central melanin production on or off switch. And MC1R is responsible for upregulating the production of melanin via downstream effects. But agouti signaling protein is this master off switch for MC1R. What this means is that when we get more ACIP expression, we get less melanin production in the cells as a whole. So if ACIP is upregulated in the gray feathers in the Anna's hummingbird, it means that it's producing less melanin by a lot. And this is totally consistent with what I'm seeing. This is totally consistent with what I'm seeing. Oh, wait, can you guys still see my screen? Oh, okay, that's good. That's totally consistent with what I'm seeing in the morphological data because the melanosomes are a lot smaller, there's fewer of them, and they're a lot more irregularly shaped and irregularly arranged. Okay, so that was pretty exciting to me. So I can begin to answer this question, is iridescence homologous from a molecular standpoint? Because I now have this information about how hummingbirds are regulating their iridescence. And there's data from two other species from studies that I did not do myself, but from a mallard and from a superb starling. So in the mallard study, they compared iridescent green feathers and pigmented brown feathers. And in the starling paper, they compared iridescent blue feathers and pigmented red feathers. So in the Anna's hummingbird, like I said, agouti signaling protein was upregulated in non-iridescent feathers. There was also this gene DCT that I haven't talked about yet that was upregulated in non-iridescent feathers. The important part is that these are all melanin synthesis genes, so they're involved in the melanin regulation pathway. But in the duck and in the starling, even though they are very different birds, they do very different things and they're not closely related to each other. They're actually using the same exact genes, tyrosinase and tyrosinase related protein 1, to regulate iridescence or non-iridescence in their feathers. So what this is telling me is that there might actually be independent evolution of the molecular pathways involved in producing iridescence, in addition to independent evolution of the morphology. So you might be wondering, is this because the duck and the starling have really similar melanosome morphologies? And we'll get back to that. And the answer is no, actually. The hummingbird and the starling are actually much closer morphologically to one another, even though it's the duck and the starling that have much more closely related genes that they're using to regulate their iridescence. What I wanted to point out here was just the melanin synthesis pathway, where hummingbirds are using agouti signaling protein to turn off melanin production, where the duck and the starling are using tyrosinase and tyrosinase-related protein 1 to increase melanin production to create their iridescent feathers. So even though the duck and the starling are turning on iridescence through their genetic mechanisms, it seems that hummingbirds are turning off iridescence using their genetic mechanisms. So they're sort of doing the reverse. And this is why I think that hummingbirds are actually expressing iridescence constitutively in their feathers, which means that they have no choice but to produce iridescent type melanosomes, which they then have to turn off using the agouti signaling protein. So in that way, hummingbirds are born to shine. There's some evidence in a similar group of birds called the cuckoos, where in this 2023 paper, it was demonstrated that some cuckoos, like this running cuckoo, this running kua, uh, that these birds, even though they don't have iridescence, use the iridescent type melanosomes to produce their pigmented feathers. Okay, I think I think that's the talk. I think that's everything. Okay. Wow. Okay, that was exciting. I have a lot of people to thank, including my advisors, some collaborators from the Field Museum, the Burke Ornithology Collections team, some lab mates that helped me in the field, and the Seward Park Audubon team that also helped me in the field. And of course, my generous, gosh, I almost said sponsors, but they're not really sponsors, are they? They're funding bodies that help me fund my research. So thank you very much, including to the SSE. Okay, I think this is the last round of questioning. I did go over time and I apologize for that, but I'm happy to stick around and answer any questions if you guys have them.
Yeah, we're slightly over time. Maybe we'll go another 3 minutes and till 05 after and uh close out the seminar. But a great talk, Yasmin. Really enjoyed it.
Thanks.
And there are people who are clapping here, so.
Yay.
I had a question about the second part, actually. Although the third, I really enjoyed the third part. Um, with crows being glossy, what do we know about like jet, magpie have iridescence, and are they more closely related to crows then than jays are? I was wondering if the crow gloss example was another gloss nested within iridescence, which was a similar pattern to what you saw in your group.
Yeah, that's a really good question. To answer that particular question, we would have to look at the phylogeny of the group, which I think is what you were implying.
Yeah.
And I don't know off the top of my head what the Corvid family tree looks like. So I'm sorry I can't help you.
No, it's okay. I was wondering because you did mention how your results may not be applicable to other parts of the bird tree, and I
Oh
was trying to
yeah.
think about how where crows were. Um,
I think the lesson at the end of the day is that historical contingency matters a lot for iridescence because it evolved so many times, because it evolved in such disparate groups. So it seems that iridescence is a useful thing to have, and so a lot of birds just develop it independently.
and I just have one, unless other anyone else would like to go, please interrupt me because of asking too many things. Um, but I know for the hummingbird phylogeny, one of the earliest diverging lineages are these Faithornis hermits,
Mm,
and they lack iridescence. So I'm curious if you get some samples of those, if they, or any non-iridescent hummingbirds, for example, Caligina, you had several Caligina species. There's one Caligina, which is just all brown, at least to our eyes. I'm
mm,
wondering if those ones that have no iridescence are turning ASIP off all over, or
mm.
if they have they're more like the duck turning something on or something like that.
Yeah, I'm gonna Google Caligina
Hmm.
after
Bonzi
this,
Inca. Hmm.
but I'm almost certain that all hummingbirds have iridescence somewhere on the body, whether or not it's like very prominent. They should have like a few green feathers on the back at least.
Oh, okay.
Yeah.
Yeah, it's just to my eye, I guess. Uh,
Yeah, but that is like a really good question about like what melanosomes they're using in that particular case. Yeah. I know, right? So many
Just a
questions.
lot of birds to think about. A lot. All right, guys. So, thank you all for staying with us till the end here. Really enjoyed Yasmeen's talk, and we will see everyone here next week for Greg's seminar. Thank you so much.
Bye, everyone. Nice to see you
Bye,
all. Thank
guys.
you.
Thank you.